Let's discuss about the reproduction in bacteria. In bacteria, the reproduction is mostly by means of asexual type. So asexual method is the type of reproduction where there is no use of gametes, means no use of gametes. And this in bacteria we find sexual reproduction present, but it is a sexual recombination, that is recombination of genes present. No gametes are involved. No gametes are involved. They don't find any gametes present. And the sexual reproduction is not the true sexual mode of reproduction. So true sexual reproduction is mostly absent. Let's start with asexual reproduction first. So asexual reproduction is by means of spores called as sporangiospores. For example, that the bacterial produce spore and this is called as sporangia. The sac-like structure is called as sporangia, and inside sporangia the spores are produced, and this spores is called as what? Sporangiospore. Under favorable conditions, the sporangia burst and the spores are released out to give rise to a new individual. The next type of asexual reproduction is by means of oidia or conidia. In conidia, same thing. The conidia produce in a chain like manner, and these individuals are called as conidia. And also oidia, also we find this type structure oidia present, and oidia mostly present in sugary medium. When the condition is highly sugar is present, ample organic matters are present, oidia reproduction do takes place. And conidia reproduction also takes place in certain favorable conditions. Got it? Okay. And under unfavorable condition, adverse condition, endosperms are produced. Means here what happened, the spores are produced outside the body. Here the spores are produced outside the body. So sporangiospore, conidia and oidia, they all are exospore. They all are exospore. But in endospore, we find the spores are produced within the body. Inside the bacterial cell, the spores are produced. So it is called as endospore. Move on to the first mode of reproduction, that is by means of cell division, that is called as binary fission, and it is the common method of reproduction. And binary fission is a type of reproduction which takes place by means of cell division. So here what happens, single cell, they divides to give two daughter cells. And this type of cell division is called as amitosis. Amitosis means no spindle fiber is involved. No spindle fibers are found because it is naked DNA. And also it is sometimes called as direct cell division. It is called as direct cell division. Got it? So by this, two daughter cells are produced, and each daughter cell then develops into parent cell, matures into parent cell, and also they start dividing to produce many daughter cells like this. And as a result, multiplication, many number is produced. So by binary fission, bacteria multiply itself. This is the most common method. So one bacterial cells divide into two daughter cells as we have discussed, and spindle fiber is normally absent as we have discussed. And also during binary fission what happened, multiplication of large number of bacteria takes place, a large population. So many bacteria are produced within a few seconds of time. The few seconds of time many bacteria are produced, because mostly in E. coli 20 minutes taken for bacteria to divide. So in 20 minutes we'll find two bacteria daughter cells. In the next 20 minutes we'll find many bacteria goes on dividing. So like this within a few very short period of time, rapid multiplication of bacteria takes place. Growth, when the population increases, when the number of bacterial cells increases, then the drawbacks. What is the drawback? The growth stops. The growth slows down. Why? The shortage of space. Not only shortage of space, lack of nutrients. Nutrients are insufficient for the growth. Not only insufficient nutrients, accumulation of waste materials also prevent the growth. And also certain bacteriophages directly destroy bacteria. Matlab means what? Now what is bacteriophage? A virus infecting bacteria. Virus infecting bacteria. So virus infecting bacteria is called as bacteriophage. So bacteria are destroyed. So these are the drawbacks in multiplication and the growth of the bacteria after binary fission. So binary fission normally occurs under favorable condition, but we find certain drawbacks are present. Now let's move into steps of the binary fission. Replication of DNA first takes place. As we know that bacteria have DNA present, naked DNA present. And during the first step, the DNA replicates. The DNA duplicates into two daughter DNA. Then the mesosomes are produced. So what happened? Now we have one mesosomes initially. But at the time of after division of duplication of DNA, the cell membrane produce two mesosomes. So two mesosomes takes place by membrane synthesis. So by membrane synthesis, two mesosomes are produced, and each mesosome will attach with the respective DNA, its respective DNA. Clear, got it? And after that, what happens? The wall formation takes place, and a cross wall takes place to form two daughter cells. So let's move. So this is the plasma membrane, this is the DNA present. So the DNA duplicates into two, and here single mesosomes are present, and here the mesosome duplicates into two, and each mesosome now joins with its respective DNA, and each mesosome now separates the respective DNA to the respective side or respective pole. So both the DNA are moved towards its respective side, and then a groove takes place, and cross-wall deposition of chemicals, minerals takes place, and the wall is forming between them, and it is being separated, and we find two daughter cells are produced. So these are the steps of binary fission. So DNA duplication, DNA replication takes place, as we all know that the DNA of bacteria is the circular DNA and is double stranded. So when DNA replication takes place, first it converts to single stranded form, simultaneously convert to single stranded form. So this is single stranded strand, this is single stranded. So now the DNA will duplicate, new daughter DNA will produce. So this method looks like the theta appearance of Greek. So this model of DNA replication is called as theta model of DNA replication. So replicating chromosome appears like a Greek theta model. Yes, so it is called as theta model. This model is called as the theta model of DNA replication. Mesosome duplication takes place, as we have discussed. Let us suppose this is the parent mesosome present, and this is the cell membrane. So here the cell membrane synthesizes to produce two mesosomes. So two mesosomes are produced by the division of the cell membrane. So mesosome divides by membrane synthesis. Yes. And chromosome move to the daughter cells, as we have discussed. And after that, cross-wall formation takes place. As we all know, from the periphery, the deposition chemicals take place. Already the chromosome and the mesosomes are separated, and this nature is called as the plasma membrane. The cross-wall starts. Not only cross-wall, deposition of chemical takes place to produce cell wall. And initially first they produce cell membrane. So first cell membrane produced, then deposit of wall chemicals takes place to form a cross-wall synthesis that will separate the two daughter cells into individual daughter cells. So new wall materials are deposited on it. Septum and cross-wall synthesis occur. So as a result, two daughter cells are produced. Now let's move into the next asexual mode reproduction, that is called as endospore. Endospores are formed in bacteria like Bacillus. In Bacillus bacteria, Rhodsia bacteria, the endospores are produced within the bacteria. So these are called as endospore. And the endospore is produced during unfavorable conditions, either at the center or at the terminal. And it is produced within the bacteria, inside the bacteria, so they are named as endospore. So endospores survive in adverse conditions. Yes, they can survive in adverse conditions. They can withstand highly adverse conditions. So they contain resistance bodies. These are sometimes called as resistance bodies that can withstand adverse conditions. Endospore may be spherical, may be oval cell, may be terminal, may be central. And they contains a special chemical that is called as calcium dipicolinic acid that provide resistance to the endospore. And this calcium dipicolinic acid, at the time of unfavorable conditions, they help in water absorption. So water absorption takes place at the time of unfavorable condition. Let's discuss. At the center, we find the nuclear materials are present, and proteins and DNA, RNA all are present. Then we find what? Cytoplasm present, DNA, RNA present, yes. After that, we find what? A wall is present. So here we find the DNA, RNA present. Then we go for a spore wall. Membrane is present, then spore wall is present. Let's move. So here what happened? The DNA, RNA present. The proteins are present. We find a core wall. The membrane is present. Inside the cell membrane is present. Then we find a core wall is present. Then cortex is present. This is the cortex layer. After cortex, the spore coat is present. This is the spore coat present. And then exosporium is present outside. So this is the structure of the endosperm. And when the endosperm matures and favorable condition arises, what happens? That the endosperm ruptures. The endosperm ruptures and gives rise to the new bacteria. Release the new bacteria. So we came to know that the cortex present. The cortex is made up of peptidoglycan and calcium dipicolinic acid. Very important. The cortex part is made up of calcium dipicolinic acid and peptidoglycan. and spore coat is surrounded by the cortex. First nucleus present, cell membrane present, spore coat present, then we find the cortex present. Then endosperm is highly resistant to desiccation, to chemicals, to unfavorable conditions, that is, that it accumulates protoplast at the center. Protoplast separates the rest of the cells, means the protoplast comes and lies at the center. Here we don't find any protoplast present, and after that we find the formation of endosperm. For example, suppose the vegetative cell is present, so endosperm start formation either from the terminal or from the center. So accumulation takes place from the center or from the terminal, and when the spore starts formation, it forms spore coat, and after spore formation, the endosperm then they breaks, this spore coat is broken and released, the new bacterium comes out. So this is the endosperm produced, and the endosperm germinates by breaking the spore coat and giving rise to the new individuals. This is how endosperm germination takes place. So endosperm can resist high temperature. Above 100 degrees centigrade it can tolerate above 100 degrees centigrade, and endosperm is unharmed during pasteurization. What is pasteurization? Heating any liquid at 60 degrees centigrade for 30 minutes is called as pasteurization, and here endosperm can withstand this pasteurization technique. Also, the wall is very thick, they are impermeable to water. The spore coat is impermeable to water, is very thick, so they helps in preventing from mechanical pressure. And low water content increases the resistance, and low metabolic activity also increases its survival. Yes, they have less water content and they have less metabolic activity, so they undergo a resting stage, they undergo dormant stage. So in the dormant stage, the survival activity increases because the metabolic activity becomes very low. And after that, when the endosperm starts going for germination, this calcium dipicolony complex present on the cortex wall, they starts absorbing water under favorable condition. So at the time of germination, that starts absorbing water with the chemical present, that is called as calcium dipicolonic acid present in the cortex. This endosperm then becomes metabolically active and they breaks the dormant stage, and the thick spore coat ruptures, giving rise to the new endosperm. So germination of endosperm, that means cell, new endosperm rises, a new bacterium arises from the endosperm. So one endosperm will produce one bacteria. So one endosperm regarded as a one parent cell. So one endosperm will give rise to single bacteria. Then in the next class, the next section will study about the sexual reproduction. Okay, let's start sexual reproduction. So here sexual recombination, it is not called as sexual reproduction, it is sexual reproduction because no gametes are involved. So bacterial sexual reproduction is primitive. Why? Now no gametes are involved. No gametes used. What happens? Now gene recombination or gene transfer or recombination takes place. So gene transfer or recombination takes place. So this type is called as sexual recombination. It is a primitive form of sexual reproduction. So let's discuss. So in bacteria, the gamete formation of fusion is absent, but there is a fusion of genes takes place, and genetic recombination occurs through this DNA exchange or by DNA recombinations. So methods of DNA recombination, there are three basic methods present. One is called as transformations, is one method of gene recombination. One is called as conjugation, another one is called as transduction. So three methods are involved in sexual recombination. So together we can call them as what? It's the genetic recombination, where the genes are involved in this transfer of characters from generation to generation. Let's move to methods of genetic recombination. So three methods, for example, transformation. So when we move into transformation, in transformation what happens? Suppose this is the donor cell and this is the recipient cells. The donor cells will release its DNA content to the surroundings, and the recipient cells will absorb the DNA components, and these DNA components is integrated into the recipient cells and convert the recipient cells into a donor form cells. This is one type of genetic recombination. Another genetic form of recombination is where the donor cells transfer the DNA or the genetic materials into the recipient cells with the help of a tube that is called as conjugation tube. This conjugation tube, and by this recipient cell converts into a donor forms only by gene recombination. Another one is transduction, where the bacterial DNA is incorporated into the bacterial DNA, and bacterial DNA gets accumulated and they forms a new combination of genes, new combination of characters, and the new bacteriophage so formed contains bacterial DNA and viral DNA, and they again go and transfer the DNA to the other, and this method is called as transduction method. So let's discuss one by one. Transformation. So Griffith, scientists discovered the process of transformation in the year 1928. He experimented on two different bacteria, Diplococcus and Streptococcus pneumoniae, that causes the disease called as pneumonia. And here he used the donor and recipient cells. And the donor and recipient cells never come in contact to each other. But the recipient cell is converted into a donor form. How? Let's move. So donor release the DNA fragment. For example, these are the DNA fragments present in the donor. The donor release the DNA fragments, and the recipient cells, these are donor cells. The recipient cells absorbs the DNA fragments. They absorb the DNA fragments. So here the recipient cells will absorb the DNA fragments and convert into the donor-like structure. So this method is called as transformations. So recipient cells actively take up the DNA of the donor cells, and this nature is called as competence. The ability to uptake the DNA is called as competence. So recipient cells are always competent in nature. What are the strains of pneumococcus? There are two different states. One strain where we found a capsule is present. So the bacteria having capsule is called as capsulated, and they are very protective in nature, and they are pathogenic. And so they are pathogenic. They are virulent, pathogenic. And other bacteria where R2 strain where no capsule is present. Where no capsule is present means they are non-pathogenic. Means our WBC antibodies will kill this bacteria. So they are unable to infect. So R2 strain is non-virulent, non-disease causing. S3 strain is disease causing bacteria. So these two strains of pneumococcus are taken in the experiment. So Griffith used both the strains experimentally, and he injected one by one into the mice. So in his first experiment, he injected this into a mice, and the mice causes this because S3 bacteria causes infection. So it causes infection, so it causes death. Second, R2 bacteria, when injected, it never causes infection because so no kill of the mice takes place. But when he used heat killed S3, the heat killed S3 when injected, so no infection. No infection takes place because the capsule is being destroyed by heat. So when heat is applied, the capsule of the bacteria is destroyed, for which they are unable to infect. So when again what he did, he mixed the heat kill and R2 strains. So this one also non-infective, this one is also non-infective. When he used both non-infective, still he find that the mouse dies. So he was a bit confused that both are non-virulent. Heat killed and R2 are non-virulent. Then why it causes death in the mouse? So experiment it is proved that the R2 bacteria contains the virulent DNA inside it, and this R2 bacteria converts into S3 strains, helps in formation of capsule, and causing death. Means, for example, this is R2 bacteria. When the DNA of the S3 is incorporated, so this DNA have the information, so it forms the capsule. And as the R2 is converted to S3 by the formation of the capsule, so they becomes infective. So Griffith concluded that the transfer of certain matters takes place, and he concluded the principle is called as transforming principle because the transforming materials from S3 to R2 causes the death of the mouse. So here what happened in the first step, S3 is injected in healthy mouse and it causes death. RT is injected to the healthy mouse, no death, mouse suffered no infection. Heat killed S3, means the capsule is being removed out by heat killed, but its DNA is injected. When the DNA is being injected, mouse survived. But R2 living and S3 living, when both are injected, we find the healthy mouse dies. So what is the reason behind it? The reason is that this R2 is converted to S3 by competent of the DNA, and it forms a new material that is called as capsule, and this capsule causes infection in the healthy mouse and causing death. This is the main reason. So here what happened, the S3 DNA is being transported into the R2 and converting R2 into S3 form, and which causes the death. So the living mouse S that causes death. So these are the S colonies formed. This the R colony is formed. Here heat killed. When heat killed means no capsulated bacteria are found, is not collected, no colonies are isolated from the tissue because already we have killed it. But in the next stage where R and heat killed, we find that the DNA is incorporated into the R and we find a new bacteria. Here some modified R2 are present that causing disease and certain R are present. For example, suppose we have taken 100 R2. After transformation, we find 50S and 50 R2. So this 50S that convert into R2 convert to 52 will cause the death, will cause capsule and causes death. So this is how transformation takes place. So this experiment was again conducted by Avery, MacLeod and McCarthy. And they experimentally proved that yes, Griffith was right. In the year 1944, they introduced that yes, the DNA is the actual genetic material that helps in transformation of information and helps in formation of new capsule in the non-capsulated bacteria and causing diseases. So DNA of the dead S3 is transformed to R2, causing and converting R2 to produce the capsule. This capsule causes the death of the bacteria. So this experiment proved that DNA is the genetic material which is transported and it gets recombined with R2. Suppose this is R2 and S3 is incorporated. So R2 and S3 are incorporated. So here this is called as transformation or it is called as genetic recombination. Both the genes are recombined. So it is called as genetic recombination. So it is one type of sexual reproduction. Now let's move to the next method of sexual reproduction or sexual recombination, that is called as conjugations. So what do you mean by conjugation? Conjugation means union. Conjugation means union. So the conjugation was given by the scientists Lederberg and Totem in the year 1946. So according to conjugation, they used the bacteria that is called as E. coli. So this process was experimented on E. coli bacteria. And they found that the DNA is transferred from the donor cells to the recipient cells by the process of conjugation and with the help of conjugation tube. With the help of conjugation tube. And also the conjugation tube is formed by the help of F plasmid of F factor. Got it? So F plasmid is the fertility factor. Suppose any bacteria, suppose the bacteria having the plasmid, then this bacteria will definitely produce the conjugation tube. If this is the bacteria present without plasmid, no plasmid, so no conjugation tube. No conjugation tube is formed. But in this particular bacteria, as the presence of plasmid is present, so conjugation tube is being produced. So conjugation tube is produced. So the bacteria having a plasmid or the fertility factor plasmid will produce the conjugation tube to transfer the DNA content. So the F plasmid converts F minus without plasmid, F plus having plasmid. For example, suppose this is having plasmid and this is the DNA. For example, this is only a DNA. So this is called as F plus and this is called F minus. So they will don't. So this acts as the donor. So F plus acts as the donor. F minus acts as the recipient. As F plus contains the plasmid, F plus contains the plasmid, minus don't have the plasmid. There are two different forms of bacteria on the basis of presence and absence of plasmid. So plasmid replicates during conjugation. Only plasmid present helps in formation of conjugation tube. Let's discuss. For example, suppose this is the plasmid present, and here what happens, as we have discussed that this bacteria contains the plasmid. So as F plasmid present, so conjugation tube is present. Plus here no plasmid present, no conjugation tube. So F minus, no conjugation plasmid, F plus with. So this conjugation tube will at the time of union, this conjugation tube will extend and attach with the recipient bacteria, and they will help in dissolving the layer, common layer of recipient bacteria by enzymatic action. So a common passage is formed, and this common passage, the plasmid, one of the plasmid replicated, and the replicated plasmid moves in the recipient cells, and this recipient cell is getting the plasmid becomes F plus, and this becomes also F plus. Initially it was F plus and it was F minus. And let's move to the next type of plasmid, that is called as HFR cells. What are HFR cells? For example, suppose this is the bacteria present and this is the plasmid present, and this is the bacteria present without plasmid. So here we call them as HF, F plus or F minus. But here what happens, these plasmid gets accumulated or integrated with the DNA. So when it gets integrated with the DNA, suppose the DNA having the plasmid is called as episome. Is called as episome. Got it? And this episome will induce this bacteria to produce two type of DNA. And again return back to the plasmid. So this is the duplication of DNA. This is one DNA, this is one DNA. Got it? Next what happened? As the plasmid is present, they will suddenly produce the conjugation tube due to presence of plasmid and two DNA present. They gets attached with the recipient cells, that is F minus cells. So here what happened? In the next stage, as usual, one of the DNA will transfer into it. So one of the DNA will find here plasmid, only one DNA present, here both the DNA gets attached mixed. So this is the modified form of DNA present in F minus. As episomes is getting attached to this particular cell, so these are called as high frequency FR, high frequency recombinations. So here high recombination frequency takes place that induce the DNA to duplicate into two forms, and one of the DNA gets transferred into the F minus, and converting this F minus is as is converted into a modified form. So this is how in HFR plasmid integration do takes place. So here such cells are called as HFR cells, high frequency recombination cells. So high frequency recombination cells, high recombination frequency. So high frequency means there's a factor present. They will convert this DNA, they duplicate the DNA, and the duplicate moves to the other DNA. This is the conjugation process. So in the conjugation process, the DNA transfers occurs through a conjugation tube. Yes, I have discussed the occurs through the conjugation tube. And efficiency depends on episome. If episome is present, maximum episome takes place, maximum conjugation takes place. And gene transfer depends upon the contact time. If the contact time between two bacteria, suppose this is the bacteria and the contact time is more, then maximum conjugation, the maximum transfer takes place. If the contact time between the two bacteria F plus and F minus or HFR and F minus is less, then less conjugation process will take place. So here what happened, let's go into the conjugation process just we have discussed. That is, for example, this is the plasmid present, this is the DNA present, and here this is they don't have plasmid, so F minus. In the replication, what happened, the plasmid replicates into two. One of the replication plasmid moves into the daughter cells, and this F plus initially present, now this F minus is converted, initially F minus converted F plus. So this is how transfer of plasmid takes place. So here plasmid takes place and convert F minus to F plus. But in HFR what happens? And in HFR the plasmid is present. The two DNA are present with the help of plasmid, and one of the DNA is incorporated into the daughter cells. And in the daughter F minus we will find two DNA present. So this F minus is a high recombinant virus, a bacteria is a high recombinant virus bacteria present. Okay. Now discuss about the transduction. So what is transduction? Transduction is the transfer of viral gene to bacteria and then to other bacteria and then to other bacteria, to bacteria and then to other bacteria. Got it? Means bacteriophage. Viral DNA genes means these are the bacteriophage. This is the bacteriophage present. Got it? So let's move. So transduction transport DNA using bacteriophage. What is bacteriophage? A virus infecting bacteria. And it was given by Zinder and Lederberg in the year 1952. And it is experimented on Salmonella typhimurium causing the disease typhoid. E. coli was experimented on conjugation, and Diplococcus pneumococcus bacteria helps for transformation experiment, and the Salmonella typhimurium is used for transduction experiment. In a transduction experiment, normally what happens, the bacteriophage present. It's the bacteriophage present. This is the bacteriophage present. This bacteriophage and is the viral DNA, bacterial DNA present. So this bacteriophage dissolves the wall of bacteria, release this viral DNA. This phage DNA moves, and inside the phage DNA, what happens? The phage DNA either it replicates by taking the DNA polymerase enzyme of this host cell, it replicates, and this replication then breaks the DNA of the host, and this DNA is broken down and is integrated with the viral DNA, and this viral DNA again forms this capsid layer and tail region, and here a new capsid is formed, new bacteria, viral bacteriophage is formed, and they dissolve the layer of the host cell, and so they causes lysis. Breaking down of bacteria is called as lysis, and the new bacteria again results and gets attached to the new fresh bacteria. And in some case, and this phase is called as lytic phase, where lysis takes place. What is lysis? Now dissolving the host bacteria, killing the host bacteria, releasing the new bacteriophage, a new modified bacteriophage is called as lytic phase. But when we move on to the lysogenic phase, here what happens? Normally, this phage DNA, they integrated with the parent DNA, the host DNA, they gets integrated with the host DNA. After integration, they helps in replication of this host DNA. And after replication of this DNA, they then causes division and forming this bacteria. And this bacteria is of one type, this bacteria is of one type. Here, this phase is a lysogenic phase where the host is not killed. The host cell is not killed, but it is multiplied. So in lysogenic stage, we will find the host cell, the bacterial cell is not destroyed or not killed. In lytic phase, we will find the bacterial cell is destroyed and only the bacteriophage comes out. Here what happens? Now here, the bacteria comes with the integrated form of DNA and called as lysogenic phase. So lysogenic bacteria, viral DNA integrates in the bacterial DNA, and such bacteria are called as lysogenic bacteria, yes, and integration takes place. Okay. But in lytic cycle, what happens? The phage participants multiply inside the host cell and kills the host cells and release new phages to the outside. So in the lytic phase, the host cell is destroyed or killed. But in faulty phase, sometimes there is a faulty phase present where some phages carry bacterial DNA fragments, and these phages, for example, suppose this is the phage present, the viral DNA present and some bacterial DNA present, and they move to the other bacteria and release the bacterial DNA to the other bacteria, and this bacterial DNA integrates, and this is called as faulty phase of DNA. This in nature is called as faulty phases of the phases of DNA. So faulty phases infect another bacteria, yes, and release the bacterial DNA enters into it and causing a new. So all this experiment we find what? Transfer of genes takes place or transfer of DNA takes place. So in this sexual recombination is nothing but it is a transfer of genes from one bacteria to other bacteria. Yes. Then we move to types of transduction. What are types of transduction? Generalized transductions means transfer any bacterial DNA. So in this form, any bacterial DNA can be transferred. Examples T4 phase. Specialized transformation, only specific DNA region of the bacteria is being transferred. So specific region of the bacteria is transferred, specific region. And here any DNA phase is being transferred, difference. And lambda phase carry specific bacterial genes. They never carry all genes. Specific bacterial genes are carried. Abortive where no DNA is being carried out. If no DNA is integrated or carried to any bacterial genome, it's called as abortive transduction. So no transduction takes place in abortive. So lambda phase is present, one kind. Abortive phase is present, another kind. And also we find specialized transduction and generalized transduction. Specialized transduction, where only all types of DNA can be integrated, T4 phase. And generalized transduction is special transduction, only special DNA region is being integrated. Whereas in lambda, specific bacterial genes are integrated. Abortive, no DNA is integrated into bacterial cells. So this is all about the types of transductions.